In this video, we're going to go over the pentose phosphate pathway. Now, all throughout glycolysis and biology, we've learned that ATP is the cell's energy currency. It allows us to do stuff. But ATP isn't the only energy reserve the cell has. It has a secondary currency in reducing, that is, reducing elements. We can get energy from that. And to harness that energy, we're going to make NADPH. Note, NADPH and NADH are not the same thing, and enzymes will not just use either or. They will only use one of them. They are not interchangeable. NADPH is used for reductive biosynthesis. That is, we'll make stuff and we'll be reducing molecules when we do that. And NADPH is generated by oxidation of glucose 6-phosphate via an alternative pathway to glycolysis. That alternative pathway is called the pentose phosphate pathway, or the hexose monophosphate shunt. Now, any tissue that is heavily involved in lipid biosynthesis, this being your liver, mammary gland, adipose tissue, and adrenal cortex, they're going to have a lot of these pentose phosphate pathway enzymes, and they're going to be the ones carrying out this reaction. And actually, 30% of all glucose that is oxidized in the liver will do via the pentose phosphate pathway rather than just straight up glycolysis. So here's the overall pathway. We don't need to focus on every single step right now. We'll be doing that a little later on. We're just going to look at the overall picture of the pentose phosphate pathway. So the overall reaction is that 3 glucose 6-phosphates plus 6 NADP plus 3 water will go to 6 NADPH plus 6 hydrogens plus 3 carbon dioxides plus 2 fructose 6-phosphates and 1 glyceraldehyde 3-phosphate molecule. So that's overall what we're doing. And this is done in 3 major stages. Stage 1 is oxidative reactions, which will give us NADPH. And stage 1 is the only place in the pentose phosphate pathway that we make NADPH. And we're also going to make ribulose 5-phosphate, RU5P. And the overall mechanism for this is, our reaction for this is 3 G6P plus 6 NADP plus 3 water will make 6 NADPH plus 6 hydrogen plus 3 carbon dioxide plus 3 Ru5P. Stage 2, this is reversible, and it's an isomerization and epimerization, transforming our Ru5P to ribose 5-phosphate. And so if you're trying to figure out where am I on the graph right now, I'm actually right here. So stage 1 was getting to the top. Stage 2 is down here. And the overall equation for this is 3 RU5P makes R5P plus 2 XU5P. Stage 3 is reversible again, can go both ways. It's carbon-carbon bond cleavage, and it's a formation reaction. It's converting 2 molecules of XU5P right here, 1 molecule of R5P right here, and you're going to make 2 molecules of F6P and 1 molecule of glyceraldehyde 3-phosphate. So that's the overall picture of the pentose phosphate pathway. Now we're going to get into each of the steps and see how they work. All right. So our first stage is oxidative reactions producing NADPH. So glucose 6-phosphate is your starting point of the pentose phosphate pathway. And this happens from hexokinase working on glucose or glycogen breakdown. So you'll make G6P from either glucose or glycogen. So step 1, you take your glucose 6-phosphate, and your enzyme is glucose 6-phosphate dehydrogenase. Nicely named, tell us what it's doing. And what's going to happen is that you have an NADP, you're going to form NADPH. Notice how weird these arrows look, how there's 3 lines. This is saying that this is non-reversible. This is very strongly favored towards the right. Our enzyme glucose 6-phosphate dehydrogenase is inhibited by NADPH, strongly inhibited. So this pathway, the pentose phosphate pathway, will only really go if you need NADPH. And what you're going to do is you're going to take a hydride ion from glucose 6-phosphate, C1, to make 6-phosphogluconate delta lactone, which is this molecule right here. Step 2, you're going to take this 6-phosphogluco delta lactone. You're going to hydrolyze it to 6-phosphogluconate, using 6-phosphogluconolactonase. So that is a mouthful. But what you're doing, taking this molecule, using the enzyme 6-phosphogluconolactonase. So it's the name of our molecule, and instead of lactone, you just add ACE. That's how you get the name of your enzyme. And you're making 6-phosphogluconate, right? So that's step 2. Step 3, we're making another NADPH. We're doing another dehydrogenase. So you can see here, when we're doing dehydrogenase enzymes, we're making NADPH. So 6-phosphogluconate is made into ribulose 5-phosphate. And the enzyme that's doing this is 6-phosphogluconate dehydrogenase. So for every molecule of glucose 6-phosphate that goes into the pentose phosphate pathway, we get 2 molecules of NADPH out. All right, so phase 2 or stage 2 is isomerization, epimerization. So we have this Ru-5P that we just made, and we're going to convert that to ribulose 5-phosphate by ribulose 5-phosphate isomerase, so shown here. An alternative fate for ribulose 5-phosphate is to turn it into XU 5-phosphate, xylulose 5-phosphate. In different enzymes, actually, the same enzyme is going to be doing this. It's different enzymes. I'm committed to its different enzymes. I just didn't read the first letter. So to make ribose 5-phosphate, you need to do ribulose 5-phosphate isomerase. To make xylulose 5-phosphate, you need ribulose 5-phosphate epimerase. So the only difference that I failed to see there is the first 3 letters of the last word. Now you might be asking yourself, how do we decide which one we're going to make? R5P or XU5P? Well, it depends what you want to do. If you are a rapidly dividing cell and you need a lot of DNA, you're going to focus on making R5P because R5P is needed for the biosynthesis of nucleotides. So you're going to be trying to make this. If you are not constantly dividing, your ratio of XU5P to R5P is going to be 2:1. So 66% of your ribulose will go to xylulose, 33% will go to ribose. And we'll see why that is as we continue with the phosphate pathway. All right, stage 3, we have our carbon-carbon bond cleavage and formation. All right, so we're going to actually not go over this right away, but just kind of take a step back. Every 3 glucose 6-phosphate molecules that entered the pathway gives us 3 Ru5P molecules in stage 1. 3 pentose are converted to 1 R5P and 2 XU5P. So of the 3 that we get, 1 goes to R5P, 2 goes to XU5P. All right, that's where we're at right now. Now, these 3 5-carbon sugars will be converted to 2 6-carbon sugars and 1 3-carbon sugar. So just so you're following along, 3 G6P molecules will give us 3 5-carbon molecules, 15 carbons in total to play with. We're going to convert our 3 5-carbon molecules into 2 6-carbon molecules and 1 3-carbon molecules. And so these 2 C6, that's a total of 12 carbons, plus 1 C3, that's 15 carbons. So you see, we're not losing carbons during this. All we're doing is we're just changing how they're attached. Right, so let's take a look at this. Our first reaction is a trans-ketolase. TPP, which we talked about in a previous video, is our cofactor here. And what we're doing is that we're taking one molecule of R5P, one molecule of XU5P. We're chopping off 2 carbons from XU5P and giving it to R5P to make cetohepulase 7-phosphate, or S7P, and glyceraldehyde 3-phosphate, or GAP. Right? Now, once we made that, our next step is a transadolase. We're going to take 3 carbons from our S7P and give them to our GAP. When we do that, we're left with a 6-carbon fructose 6-phosphate and a 4-carbon etherose 4-phosphate. So this blue box is showing that this came originally from our xylulose, and now it's been given over to make fructose. Once we have that, we take another xylulose 5-phosphate and we combine it with our F6P and we combine it with our E4P. Our F6P here, it's done. We're not going to touch this anymore. But from this, we're going to take a 2-carbon unit from xylulose 5-phosphate, give it to our E4P to make another fructose 6-phosphate, and we're left with a glyceraldehyde 3-phosphate. All right, so that's why originally we make 2 molecules of xylulose and 1 molecule of ribose, because when we're making our products, which are 2 molecules of F6P and 1 molecule of GAP, we only use 1 molecule of R5P and 2 molecules of xylulose 5P. I know that was a lot of names and a lot of carbons, but here's the overall math. Step 6. Carbon 5 plus a carbon 5 molecule combine to make a carbon 7 and carbon 3 molecule. Step 7. This carbon 7 and carbon 3 molecule get rearranged to make a carbon 6 and a carbon 4. This carbon 6 is done. We don't need it anymore. It's a product. It's gone away. We take our carbon 4 and we combine it with a carbon 5 again to make a carbon 6 and a carbon 3. So overall, 3 carbon 5s plus will make 2 carbon 6 and 1 carbon 3. That's the pentose phosphate pathway. Now, this pathway needs to be regulated because it's taking glucose that could go through glycolysis and using it to either make nucleotides or to make NADPH. So the products are either R5P, which go to nucleotides, or NADPH, which can be used for biosynthesis purposes. So our transadolases and transketolases, as we just saw, will convert any R5P that is not made to nucleotides to our glycolytic intermediates when we need to make NADPH. So if we really need to make NADPH, this R5P will be used to make glycolytic intermediates instead of just going on to make nucleotides. However, if we really need to make nucleotides, this F6P and GAP can then just be recycled, as this is what's being shown here. So R5P will be made into nucleotides. This XU5P can just be recycled to make F6P and GAP, which can go back to make G6P, which can be made to RU5P. So we can keep making nucleotides by just doing this cycle. And really the flux of how we go through this pentose pathway and the rate of NADPH is controlled by how much glucose 6-phosphate, or is controlled by the glucose 6-phosphate dehydrogenase reaction. This was the first reaction in the pentose phosphate pathway. The delta G is very negative, so it's very far away from equilibrium. And as we saw in glycolysis, any enzyme that's far away from equilibrium is a target to be regulated. And this is actually regulated by our NADP plus concentration. So the logic of this is, in the body, when you use NADPH, the amount of NADP plus rises. This will go and actually make G6PD, which is glucose 6-phosphate dehydrogenase. This will activate this enzyme, which will stimulate it to take any G6P and go down the pentose phosphate pathway and remake NADPH. You can also control the amount of enzymes you have by hormonal control, that is, decrease or increase the amount of enzyme that you actually make in the cell. That's it for the pentose phosphate pathway. Hope you could follow along with all those carbons. If not, just go back and look at the diagram and read the book, listen to me. Otherwise, I will see you next time.